Today we're going to learn about spermatogenesis. Spermatogenesis is the male counterpart of the gametogenesis part. You can see a general discussion on the gametogenesis in a previous video. So in spermatogenesis, what happens is we have a spermatogonium, which is a primitive germ cell, and that is converted, that undergoes a process, a systematic process by which it is converted into a mature and functioning spermatozoan, which is capable of fertilizing an egg or a female gamete, and undergo the process of fertilization. So this is the reason why we are learning spermatogenesis. Spermatogenesis begins at the time of puberty. Okay, this is in contrast with what happens in oogenesis, in which the oogenesis part will begin from before birth. Okay, spermatogenesis, the spermatogonium conversion to spermatozoan occurs at the timing is at the time of puberty, but in the female, the oogonium will start its division before birth. So that a distinction we will learn later when we discuss about the oogenesis part. So we are going into the details of the spermatogenesis. Where does spermatogenesis occur? Of course, spermatogenesis is a male gamete formation, so it should occur in the male gonad. The male gonad is the testis. Okay, this is a schematic of the testis. This is the site of sperm formation, and within the male gonad, sperms will form, and that will communicate through duct systems. These are called efferent ductules into this structure called epididymis. And epididymis has a head, a body, and a tail. And this is the site where the spermatozoon will mature, and that will be transported through this duct, and this is called the vas deferens. So this is the basic idea about the male gonad and the duct system. This is not at all a detailed description, but the site of spermatogenesis is within the testis. Now, within the testis, you can see lobules, and within each of these lobules, you have 1 to 4 seminiferous tubules. So these seminiferous tubules, as you can see in this picture, a seminiferous tubule is pulled out. Okay, when you actually dissect the testis in gross anatomy, you can actually remove thread-like structures from it, and these structures are called seminiferous tubules. So such a thread is thrown out of the testis in this schematic picture, and that is called a seminiferous tubule. The wall of the seminiferous tubule is a site where spermatogenesis is occurring. In the wall of this seminiferous tubule, you can see the lumen on the inside, and this is the basal aspect. Within this rectangle, this wall is expanded into the next figure. Okay, here you can see the seminiferous epithelium. This much thickness is the seminiferous epithelium. So seminiferous epithelium contains all the spermatogenic cell line by which you can see the formation of the spermatozoan on the luminal aspect. Okay, this is the luminal aspect. You can see the luminal aspect here. You can see the luminal aspect here, and this is the basal aspect. You can see that basal aspect here. Okay, so this thickness of the seminiferous epithelium is thrown out here. So let us look which are these cells that you see in the seminiferous epithelium to get more an idea. In the basal aspect, that in the B is representing basal. So in the basal aspect, you have the spermatogonial cells. Okay, spermatogonial cells. Spermatogonial cells are found on the basal aspect. Spermatogonial cells are diverse types. You can see different names here. You can see a spermatogonia type A and spermatogonia type B. Spermatogonia type A is again further divided into A dark and A pale. Okay, A dark and A pale. A dark is basically the stem cell. Okay, you can think that this is A dark. Okay, this is A dark, and A dark, what happens to A dark is A dark undergoes repeated divisions of mitosis. Okay, you can see mitosis here. So it undergoes mitosis, and it undergoes mitosis to produce again A dark. Okay, A dark type of cells, and also A pale type of cells. A pale type of cell is the more committed one. The A pale type of cell will undergo division to form the further spermatogenic cell line. So A dark is basically stem cell. It is basically sort of a reserve cell. So you can see A dark is dividing and creating more and more reserve cells. You know that spermatogenesis is basically something that begins at the time of puberty but continues throughout life. So throughout a male parent's life, you need spermatogenesis. It needs to be carried on. So you need a constant replenishing of the stem cells. So this A dark will undergo mitosis to again recreate or mitotically divide and replenish or refill the A dark stem cell population. The A pale, okay, A pale cells are more committed and they will again undergo mitosis. Again, this is a mitosis. Okay, mitosis and mitosis, the product of that mitosis is type B spermatogonium. Now, why it is called dark and pale? It is because of the color of the chromatin. A dark will have a more darker chromatin material. A pale is a lighter chromatin material. So that's why it's called dark and pale. So that's about the spermatogonial cells. So you can see that spermatogonial cells again undergoes repeated cycles of mitotic division. So altogether, mitosis from the type A spermatogonium to undergo this division mitosis almost 16 days. Okay, so 16 days of mitotic division occurs throughout the spermatogonial cell lines. A dark, a pale, and then to B. Another event that I need your attention into is when the A dark divides into the A pale cells and further divisions, you can see that the cells are not completely dividing. They are connected. They are connected by an intercellular bridge. Okay, why this occurs? You imagine this is a cell. When that cell divides into two, you know that first initially the chromosomes will split, and after that the cell, the cytoplasm will divide. It is called cytokinesis. So here the cytokinesis is not complete. So if this is a cell, that cell instead of dividing completely, the cytokinesis is not complete. You have a cell bridge that is connecting these two cells. Okay, so you may have the nuclei again formed, but you have a cell bridge connecting these two. Again, when it divides further, you can see repeated cycles of cell division. This cytoplasmic cell bridges that you see is connecting the cells throughout, and you can see that is forming an elongated chain. Okay, this is called the string of pearl appearance. So many cells, but all of them are connected by cell bridges. Okay, this is due to incomplete cytokinesis, and this is a somewhat unique feature within the spermatogenic cell line. The effect of this is that you actually have clones of cells that are undergoing division in synchrony, which are also in physical contact, in cytoplasmic continuity, and they will be undergoing the stepwise division in synchrony. So after the type B spermatogonium, again undergoes mitosis. Okay, this point is may be confused by students. This is not meiosis. This is again mitosis. The mitosis product will be primary spermatocyte. Okay, this is primary spermatocyte. So primary spermatocyte is formed by the mitotic division of type B spermatogonium. Now, as I discussed in the gametogenesis video, the primary spermatocyte undergoes meiosis 1. Meiosis 1, and in the male spermatogenesis, meiosis 1 is prolonged. You can see it is 24 days. It has a 22 days prolonged prophase and a sudden completion. Within 2 days, it completes the meiosis 1. So the primary spermatocyte undergoes meiosis 1. This is often asked MCQ. Primary spermatocyte undergoes what? Or primary spermatocyte undergoes meiosis 1 or mitosis or meiosis 2. Don't forget, primary spermatocyte undergoes meiosis 1, and the product is a secondary spermatocyte. Okay. So the secondary spermatocyte undergoes meiosis 2. Okay, meiosis 2. You can see the meiosis 2 here, and here the meiosis 2 in contrast occurs only for some hours. Okay, but the meiosis 1 is occurring, it is occurring for 24 days. So in this seminiferous epithelium, in the basal aspect, you will see spermatogonial cell population. Here you will see primary spermatocyte cell population. The secondary spermatocyte is very rare to be detected in a histology specimen of the testis because the meiosis 2 completes within hours. So it is unlikely to get it in a section. Okay, so that is meiosis 2. And after that, the product is spermatid. Okay, the spermatid again undergoes a differentiation. This is not a cell division. This is only differentiation. It is only a remodeling of the cell. The cell undergoes remodeling and structural and functional maturation and differentiation, which will make it into a functioning spermatozoa. Okay, so altogether you can convert, you can basically divide the spermatogenesis into three phases. The first is the spermatogonial phase, and you can see that the spermatogonial phase is primarily mitotic. And the end of spermatogonia, you have spermatogonium type B. The type B also undergoes mitotic division to form the spermatocyte. The next phase, so this is spermatogonial phase. The next phase is spermatocyte phase. Okay, spermatocyte phase. I'll write it here. Spermatocyte phase. This is spermatogonial phase. Okay, and the next is the spermatid phase, also called spermiogenesis. 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 This is the spermatid phase. So these are the three phases, the phase 1, phase 2, and phase 3 of spermatogenesis. The first is spermatogonial, which is occurring in this part of the seminiferous tubule, the basal aspect. Next, you have the spermatocyte phase, which is occurring more on the luminal aspect. Next, you have the spermiogenesis, which is occurring on the most luminal aspect of the seminiferous tubule. Okay, these are the three phases. Altogether, the time duration of a single spermatogonial cell to become spermatozoan will be 72 days, approximately 72 to 74 days. Okay, this is also this can also be asked as a MCQ. So this is a basic idea about the seminiferous epithelium and what are the hierarchy of the cells. Okay, the different population of the cells that you find in the spermatogenic cell line. Next, we are going into a little more detail of the seminiferous epithelium, basically to understand the environment there, especially to understand a couple of cells that are located in the seminiferous epithelium and nearby. Again, this is a section of the arc of the seminiferous epithelium. You should understand that this is the seminiferous tubule. Okay, we are seeing only the arc, this much arc of it. Okay, this much arc of it is visualized in this picture. Okay, so this is the whole seminiferous tubule. You are seeing only an arc of it. Now, this is the luminal aspect again, and this is the basal aspect. You can see the very clear basal lamina here. It is actually multi-layered basal lamina. And you can see the blue colored, the ones that are colored as blue, that is a spermatogenic cell line. These cells, I'll just change the color into, yeah. These cells that you find are the spermatogonia. This is the nuclei and this is a cell. That is the spermatogonial cells. This is the primary spermatocyte. Okay, these are the primary spermatocytes. You may get a secondary spermatocyte. And then you have the spermatids. Okay, the hierarchy will be from the basal aspect towards the luminal aspect. And these are the spermatozoan. Okay, these are the spermatozoan. So the hierarchy of the cells are spermatogonium, primary spermatocyte, secondary spermatocyte, spermatids, and spermatozoan. The secondary spermatocytes will be mostly unseen in a section because it rapidly goes, within a few hours, it transits through the secondary spermatocyte phase. So it is unlikely to find it in a section. You can see that these cells, the blue colored cells, are actually located in recesses of another type of cells, another type of cell population that you find in the seminiferous epithelium. And I want your attention on that type of cell, and that is called a Sertoli cell. I'm going to show that Sertoli cell here by outlining it. Okay, this is a Sertoli cell. I'm going to draw the outline of that cell. Okay, this cell is a Sertoli cell. You can see it has a lot of processes or invaginations in which the spermatogenic cell population is populated. Okay, you can see the spermatocytes, the spermatids, the spermatozoans, spermatogonia, all are located within recesses or pockets within the processes of these cells. And these cells are called Sertoli cells. Okay, Sertoli cells, also called nurse cells, NURSE, nurse cell, also called sustentacular cells, SUST, sustentacular cells. Okay, Sertoli cell is based on a scientist's name who is Enrico Sertoli. He found it in 1865. But recently, now the trend in anatomy is to avoid eponymous terms, but still it is very commonly used. So this is Sertoli cell. Okay, I'm going to detail a little of functions of this Sertoli cell. You can see one Sertoli cell here. This is a process of a Sertoli cell here. Okay, you can see that process, and you can see another. This is another Sertoli cell. This Sertoli cell is also giving off a process. So if I highlight this aspect over here, this is a process and this is another process. So these two processes are meeting over here. This site, the Sertoli to Sertoli cell, the Sertoli to Sertoli cell, the site of addition. This site is a location of very tight junctions. Okay, you know, tight junctions are also called zona occludens. So zona occludens are present here, which makes this entire spermatogenic cell line divided into two compartments. You have a compartment over here and you have another compartment over here. Okay, this division into two is made by this Sertoli cell contact, the sites of Sertoli cell contacts, and this forms tight junctions, and this is the reason why you have a blood testis barrier. So the blood testis barrier is delimiting the seminiferous epithelium into two territories. One territory is on the luminal aspect and another territory is on the basal aspect. You also have another term for this. This is also called adluminal compartment, and this is also called abluminal compartment. Okay, this is a little bit confusing terminology, but you can remember that ab is anything away from the lumen, so it is called abluminal. Ad is anything nearer to the lumen. You can remember this word by abduction is abduction of the shoulder, abduction is away from the midline, adduction is nearer to the midline. It's similar to that. Okay, so this is adluminal compartment or the luminal compartment. This is the basal compartment or the abluminal compartment. Now, why is this important? This is important because you know that in the previous class, in the first discussion on gametogenesis, I mentioned that when the spermatogonial cells are identical, they are mitotic products of the basic primordial germ cells of the parent. But the primary spermatocyte, once it undergoes meiosis 1 and then further meiosis 2, it undergoes crossing over, exchange of genetic material, as well as independent assortment, which is found in the meiotic cell division. Due to these two, these cell populations are genetically different from the parent. So the immune system of the parent should not cross beyond the blood testis barrier. Okay, so this much area is sort of sequestered. The luminal compartment is sort of immunologically sequestered in the parent's body. So the parent's immune response cannot fight into the luminal aspect, into the luminal compartment, due to this blood testis barrier that you find here. So the cells that are predominantly responsible for the blood testis barrier is the Sertoli cell. Okay, that is a very important function of Sertoli cell. Sertoli cell also has some other functions. For example, it is important for secreting a structure called androgen binding protein. It produces ABP. Androgen binding protein is essential for concentrating testosterone, which is the androgen, and that concentration onto the luminal aspect is important for spermatogenesis. It is also important, Sertoli cell is also important for giving structural or physical support to the spermatogenic cell line, also metabolic support by providing nutrition. It also is important. This is the spermatid cells, and the spermatids undergo a transformation into mature spermatozoa, and you have a lot of cytoplasm that will be left, and these cytoplasm will also be phagocytosed by the Sertoli cell. So Sertoli cell has a lot of functions which are going to help spermatogenesis. Okay, the Sertoli cells are very important for spermatogenesis in all these. It also has endocrine function. For example, it produces a hormone called inhibin, which is important in feedback mechanism for the pituitary. Okay. So in a lot of ways, Sertoli cells are important. Now, structurally, you can see identify Sertoli cell by identifying this nucleus. The Sertoli cell is basically touching the basal membrane, but the nuclei are sort of perpendicular to the basal lamina. Okay, these nuclei are almost rounded, but this is almost elongated and they are perpendicular. So basically, they are almost like columnar cells, right? The columnar cells that are located on the basal lamina with nuclei that are perpendicular to the basal lamina. So that is the arrangement of a Sertoli cell. This is how you can identify a Sertoli cell in a histology section. We'll come to that when we discuss histology. So that is about Sertoli cell. Now, next we are going into another cell that is important, that is called Leydig cell. Leydig cell is not found in the seminiferous epithelium. It is found outside the seminiferous epithelium. That means it is found in the interstitium. Okay, between one seminiferous tubule and another seminiferous tubule, all that area is called interstitium. So in the interstitium, you have the Leydig cell. So Leydig cell also has another name. It is called interstitial cells. Okay, interstitial cells. Interstitial cells are important. They are steroidogenic cells, and these cells will produce testosterone. Testosterone is the male, the androgen, and you can see capillaries located interstitial space of the testis, and these capillaries are important for you know that these are hormones. The hormones will be drained by the capillaries. So this is important for hormone secretion. Okay. So these two named cells are also important. I told you this produces testosterone, and testosterone will be concentrated by the androgen binding protein ABP that is produced by the Sertoli cell, which is again important for spermatogenesis. So you know that though we learn about spermatogenesis with these cell lines, you can understand that how important Sertoli cells and Leydig cells play, what an important role the Sertoli cells and the Leydig cells play in the spermatogenesis process. Okay. Now, the hormones that are acting on spermatogenesis. I told you that you have Sertoli cells here. This is Sertoli cell, and you also have Leydig cells. Leydig cells are found in the interstitium. Okay, here all you will have clumps of Leydig cells in the interstitium. So the two hormones, two gonadotrophic hormones that are important are FSH and LH. Okay, FSH and LH are produced by the anterior pituitary. The anterior pituitary is again under control of the hypothalamus. Okay, but FSH is going to act on the Sertoli cells. Okay, so Sertoli cells are counterparts of the follicular cells that you find in the ovarian cycle, which we will discuss in the ovarian when we discuss about the ovarian cycle. So the Sertoli cells are under the influence of the FSH, and LH is also called interstitial cell stimulating hormone. I told you Leydig cells are also called interstitial cells, and so it is also called ICSH. The ICSH or LH hormone will act on Leydig cells, which will produce androgens. Okay, so in this way, these two are also supporting spermatogenesis. So though we learned about the steps of spermatogenesis in 3 phases, you now understand that the Sertoli cells and the Leydig cells are important, and also it has hormonal supports, which is basically guided by the pituitary, which is under the influence of hypothalamus. This is all important in physiology, which will be detailed in physiology classes, but this is the structural substrate that you need to learn from anatomy and embryology to understand spermatogenesis. The last phase of spermatogenesis is called spermiogenesis, and that I'll consider in the next video.